Students, today we'll discuss about cell organelles. So, cell organelles are metabolically active living structures of cytoplasm, right? In cell organelle, first we'll discuss about endomembrane system. In endomembrane system, we have total four organelles. What are those four organelles? These are endoplasmic reticulum, Golgi complex, lysosome, and vacuole. These are four organelles which are included in endomembrane system. Now, why these four organelles are included in endomembrane system? Because the functions of these organelles are coordinated. How the function is coordinated? Suppose that is the structure of a nucleus, right? So outer membrane of nucleus, that is this membrane is associated with this endoplasmic reticulum. So the green structure which is shown here is endoplasmic reticulum. This is endoplasmic reticulum. The red dots on these endoplasmic reticulum are ribosome. Hence this endoplasmic reticulum is rough endoplasmic reticulum, right? So this ribosome helps in protein synthesis. Proteins are synthesized in the form of vesicles. These vesicles will fuse with Golgi apparatus. So this complex is Golgi apparatus or Golgi bodies. So these vesicles transport through this Golgi complex and reaches at this phase. This phase is the forming phase, right? So proteins are released in the form of vesicles. These are the vesicles. So this is endoplasmic reticulum, this is Golgi apparatus, and these vesicles can be vacuole or lysosome. So we can say all the four structures have coordinated functions. Since they have coordinated functions, that is why they are considered in endomembrane system, right? So in endomembrane system, first organelle to be studied is endoplasmic reticulum. Right? So endoplasmic reticulum, definition of endoplasmic reticulum. Endoplasmic reticulum is nothing but it is network of interconnected tiny tubular structure in cytoplasm. Let us draw the diagram of endoplasmic reticulum. This is the structure of nucleus. Then we'll draw outer membrane of nucleus, and these are the nuclear pore, right? So outer membrane of nucleus is interconnected with this membranous extension, right? So these are endoplasmic reticulum, right? So basically interconnected, these are interconnected cisternae of endoplasmic reticulum. These are interconnected tiny tubular structure which are spread throughout the cytoplasm as endoplasmic reticulum. Next point, it was discovered after the discovery of microscope and under electron microscope it is studied as network of reticulum of tiny tubular structure, right, which is scattered throughout the cytoplasm. Next point we'll discuss about components of endoplasmic reticulum. We have three major components of endoplasmic reticulum. Components, or we can say it is structural forms. Three type of structural forms are present. One is cisternae, second is vesicles, third is tubules, right? Now what is cisternae? Cisternae is this membranous structure. Vesicles, spherical body, right? Tubules are this branched structure is tubule. So cisternae are long, flat and unbranched structure. That is cisternae. Vesicles are oval, membrane bound structure. Tubules are irregular, branched tubes around the bounded by membranes, right? So these three are structural forms of endoplasmic reticulum. Now we have two type of endoplasmic reticulum. One is rough endoplasmic reticulum. Rough endoplasmic reticulum. It can also be written as RER. And the second is smooth endoplasmic reticulum. It is also written as SER. So two types of endoplasmic reticulum are rough endoplasmic reticulum and smooth endoplasmic reticulum, right? So what is the difference between these two endoplasmic reticulum? Rough means they are rough in appearance. Smooth endoplasmic reticulum means they are smooth in appearance. Now why they are rough or smooth in appearance? That is due to presence of a special structure on their surface. Now what is that special structure? That special structure is ribosome, right? So they have rough appearance because of ribosomes on their surface. So how ribosomes are attached on their surface? ATS type of ribosome, since it is eukaryotic cell, so type of ribosomes are ATS. ATS type of ribosomes binds by their larger subunit. Suppose a ribosome has two subunits. One is larger, one is smaller. Then larger subunit binds to the surface of endoplasmic reticulum with the help of special proteins, and these special proteins are ribofourin proteins. These ribofourin proteins are in two forms, ribofourin 1 and ribofourin 2. While they are smooth in appearance, so ribosomes and riboforeign proteins. Both are absent in smooth endoplasmic reticulum. If we talk about the structural component like cisternes, vesicles, and tubules, so rough endoplasmic reticulum is mainly composed of cisternae, right? And smooth endoplasmic reticulum is mainly composed of tubules, right? Rough endoplasmic reticulum have ribosomes, and function of ribosome is protein synthesis, right? So protein synthesis is function of ribosome. So we can say rough endoplasmic reticulum is also involved in protein synthesis. So they are more abundant in cells which are actively involved in protein synthesis. Now, what are those cells which are actively involved in protein synthesis? Example is liver cells, pancreatic cells, right? So these can be examples of the cells in which endoplasmic reticulum is more abundant. Function of smooth endoplasmic reticulum is these are concerned with glycogen and lipid metabolism. Lipid metabolism, or we can say synthesis of steroid hormones in animal cell. That is also function of smooth endoplasmic reticulum. So it helps in glycogen and lipid metabolism, then steroidal hormone synthesis. Next is detoxification of drugs. Next is detoxification of drug. Detoxification of drugs are also function of smooth endoplasmic reticulum. Next is there's one important special note. Rough endoplasmic reticulum is mainly associated or it is present near the nucleus, right? While smooth endoplasmic reticulum is mainly present towards the periphery of the cell, right? Next point about endoplasmic reticulum is modification of endoplasmic reticulum. In modification of endoplasmic reticulum, we have just one important point, that is sarcoplasmic reticulum. Sarcotome, you can remember from human physiology that sarcotome is used for muscle cells, right? So these are modification of smooth endoplasmic reticulum. You can simply write it is modified SER, right, in skeletal and cardiac muscles. What is the function of sarcoplasmic reticulum? It stores calcium and energy rich compound. Now, what is the function of this calcium and energy rich compound? It helps in muscle contraction. So, question can be asked in such a way that which organelle is responsible for muscle contraction? It can be smooth endoplasmic reticulum or sarcoplasmic reticulum, which is also modification of smooth endoplasmic reticulum. Both are same, right? Now, we'll write about functions of endoplasmic reticulum. Function of endoplasmic reticulum, it forms endoskeletal of the cell. Since it is spread throughout the cytoplasm, so it is also form endoskeletal of the cell. Second, it forms intercellular, it forms intracellular conducting system. First, we'll learn about intercellular and intracellular. Suppose we have a group of cells, right? Cell 1, cell 2 and cell 3. Inside the cell is intracellular. In between two cell is intercellular. Means in between two cell connection is intercellular, while if we talking about any component which is present within the cell, then the term intracellular is used, right? So if we are saying it is present within the cell, then it forms intracellular conducting system. Conducting system means it is involved in transport of materials in cytoplasm for one place to another, right? So that is the second function of endoplasmic reticulum. Third function of endoplasmic reticulum, since rough endoplasmic reticulum have ribosomes on it, so it is involved in protein synthesis. Protein synthesis is function of RER and lipid and glycogen metabolism or steroidal hormone synthesis is the function of SER. Muscle contraction is the function of SER. Muscle contraction is the function of SER. Detoxification of drug is the function of SER, right? So these are the functions of endoplasmic reticulum. Now what last important point which is given in your NCERT summary, that ER or mainly SER helps in synthesis of lipoproteins and glycogen, right? Other than that, it is involved in membrane biogenesis. That is not really important, right? So these are the function of endoplasmic reticulum. Next organelle of endomembrane system is Golgi complex. Next organelle is Golgi complex. Now Golgi complex was discovered by Camillo Golgi in 1898. He observed densely stained reticular structure near the nucleus. If we are talking about stain structure, then which stain is used for Golgi apparatus or Golgi complex, that is also important, right? So silver nitrate is a stain which is used for staining procedure of Golgi apparatus, and staining by silver nitrate, this method is known as metallic impregnation method, right? In plants, Golgi body are named as dictyosomes, right? In plants, Golgi bodies are named as dictyosomes. Now, just like endoplasmic reticulum, Golgi bodies also have structural forms. What are those structural forms? It can be found in the form of cisternae or tubules, vesicles, or Golgian vacuole, right? So first we will discuss about cisternae. Cisternae we have already discussed it. It is membranous sacs, flat membranous sac, which are arranged parallel to each other in Golgi apparatus, right? So dimension of these cisternae, it has diameter of about 0.5 micrometer to 1 micrometer, right? Number of cisternae in complex is variable. Variable means in general it is 4 to 8 in number, right? These cisternae are concentrically arranged near the nucleus, right? Now, most important thing, suppose that is the structure of nucleus and two phases are arranged of Golgi bodies, right? So there are two phases. One is the cis phase or forming phase or convex phase, okay? And the other is trans phase or maturing phase, which have concave surface, right? So this cis phase or forming phase is towards the It is located towards the nucleus. We'll discuss about the function of cis phase and trans phase. It is located towards the membrane, right? Now, one more important special note about these two structures. Cis phase and trans phase are entirely different structure but interconnected through cisternae. Suppose this is cis phase and this is trans phase. This is interconnected with the help of cisternae. So basically cis phase will receive the protein synthesized by RER and trans phase will release the protein modified by these network of cisternae. So that is why the cisternae or network of cisternae is present in Golgi apparatus. Next is tubule. Tubules are nothing but irregular and branched structure, just like endoplasmic reticulum, right? Vesicles are spherical or oval shaped structure. Golgi vacuoles are large spherical membrane bound structure, right? Now we'll discuss about functions of Golgi complex. What are the functions of Golgi complex? Number one, most important, is cell secretion. That is its chief function or main function. Chief function of Golgi body or Golgi apparatus. Chief function of Golgi body is cell secretion, or we can say export of macromolecules, right? Now in secretion we have three major steps. Number one, in this Golgi body will receive the proteins synthesized by RER. Suppose this is nuclear membrane and this is endoplasmic reticulum, and Golgi body, this is cis phase of Golgi body. This endoplasmic reticulum will form proteins in the form of vesicle. This vesicle will fuse with cis phase of the, that is the cis phase. This vesicle will fuse with cis phase of the Golgi body. So that is the first step of secretion. Second is, now these material are chemically modified by Golgi body, and third is it is released, or we can say secreted through trans phase. Now see, these are the three steps. Suppose this is the trans phase, so these vesicles are released or secreted from trans phase. So main function of Golgi apparatus and we'll compare this function with endoplasmic reticulum. Endoplasmic reticulum will synthesize the protein in raw form, and this is packaged and modified. Packaging and modification of protein formed by rough endoplasmic reticulum take place in Golgi apparatus. Now in this, special note is synthesis of glycoprotein and glycolipid is also the function of Golgi apparatus. So synthesis of glycoprotein is glycosylation and synthesis of glycolipid is glycosylation. So this question came in NEET, and these two terms are also important. Synthesis of glycoprotein and glycolipid is function of Golgi apparatus or Golgi bodies. Synthesis of glycoprotein is glycosylation. Synthesis of glycolipid is glycosylation, right? One more important note. Since it is used in packaging and modification of protein which is synthesized by RER, so it is also known as middle man of the cell, right? Second function of Golgi apparatus is formation of lysosome. Lysosomes are released from trans phase of the Golgi apparatus, right? So the enzymes present in lysosome are synthesized by RER. So precursor of lysosomal enzyme is rough endoplasmic reticulum, but it is packaged or modified by Golgi apparatus, and so formation of lysosome is it is the function of Golgi apparatus, or we can say collectively it is function of Golgi apparatus and endoplasmic reticulum. It also helps in synthesis of cell wall. In cell wall formation, we have also learned about that Golgi apparatus is helpful in synthesis of cell plate formation. And one more important point, in if we consider the structure of a sperm, this is head, middle piece and tail. So head is covered with acrosome, which is cap of the sperm, which contains the enzyme for fertilization or for entry into the egg cell, right? So formation of acrosome is function of Golgi bodies, or we can say acrosome is nothing but modified Golgi apparatus. That is all about Golgi apparatus or Golgi body. Functions of Golgi body is packaging and modification of protein which is synthesized by rough endoplasmic reticulum. That is the most important function of Golgi apparatus. Next, endomembrane. Next organelle of endomembrane system is lysosome. Lysosome is also known as suicidal bag of the cell, right? These are single membrane bound vesicles or vesicular structure. It is formed by packaging in Golgi apparatus. Right? Now, what are the enzymes which are present in lysosome? The category of enzyme is hydrolytic enzyme. Hydrolytic enzymes are carbohydrases for digestion of carbohydrates, lipases for digestion of lipids, and proteases for digestion of proteins. So these are the category of hydrolytic enzyme which is present in lysosomes, right? What are the function? Favorable condition for functioning of these hydrolytic enzymes. Which condition is required for these enzymes, right? It requires pH approximately 4.5 to 5. Basically, acidic medium. This acidic medium is achieved by, suppose that is the structure of lysosome, and outside medium is obviously cytoplasm. It is achieved by movement of H plus ion into the lysosome by the action of utilization of ATP molecule, right? So, acidic medium is required for functioning of these hydrolytic enzymes. These hydrolytic enzymes are also known as acid hydrolases, right? Next, most important point about lysosome, we call it as suicidal bag of the cell. Why it is known as suicidal bag of the cell? Suppose if there is any damage into the cell, then lysosome will burst. In case of any damage in the cell, lysosome will burst, and it has acid hydrolases. Now, what would be the function of acid hydrolases or hydrolytic enzyme? It will digest its own cell, its own cellular components, right? That is why it is known as suicidal bag of the cell, and digestion of its own cellular component is known as autophagy or autolysis, right? Phagy means digestion, lysis means breakdown, auto means self. So, what would be the functions of lysosome? First is it is suicidal bag of the cell, means it helps in cleaning of the cell, or mainly autophagy or autolysis. lysis, right? It also helps in extracellular digestion. Next function of lysosome means this autophagy or autolysis is termed as intracellular digestion, digestion of components within the cell. Digestion of component outside the cell is extracellular digestion, and autolysis is termed as cellular digestion, right? It is also known as squenches of the cell because it helps in cleaning process, right? So these are the function of lysosome. Next component of endomembrane system is vacuole. Vacuole is also single membrane bound organelle, right? The membrane present in vacuole of plant cell is tonoplast. The name of the membrane is very important. It is bounded by tonoplast, which is a single layered structure, right? Now, in which cell vacuole is present? It is present in plant cell. In plant cell, there is large central sap vacuole is present. It is large means very large. It can occupy 50 to 90% of the cell volume. One centrally located large vacuole is present in plant cell. In animal cell, either it is absent or if present, then it is scattered and small vacuoles, right? Membrane of vacuole is tonoplast. Now, suppose this is a plant cell and that is vacuole and this membrane is tonoplast, right? Movement of solute will move from cytoplasm towards the into the vacuole, and this movement is active transport, or we can say against the concentration gradient. Now, why the movement is against the concentration gradient? Since in cytoplasm solute particle is less and in vacuole solute particle is more, because vacuole is the sap vacuole stores. Function of vacuole is storage of waste material. Now, which waste material or waste substances are stored in vacuole? It is water, sap, excretory product, it is sap, it is excretory product, and other material which is not useful for the cell, right? So these are stored in the sap vacuole, and concentration of these molecules are, these component are high in high in the sap, and it is low in the cytoplasm. So movement of these solute particles from cytoplasm into the vacuole is against the concentration gradient, means from low concentration to high concentration, right? So in simple language we can say tonoplast facilitate the transport of number of ions and other material from against the concentration gradient, right? Against the concentration gradient means low concentration to high concentration, that is from cytoplasm to the vacuole, right? Next is type of vacuole. How many type of vacuoles are present in a cell? Different type of vacuole. So it can be food vacuole, which is used for engulfing food particles. Then it is contractile vacuole, and it is sap vacuole. Sap vacuole is present in generally plant cell, right? For storage of excretory product. Contractile vacuole is for osmoregulation, and food vacuole is for digestion or obtaining nutrition. So osmoregulation is example of osmoregulation or contractile vacuole present in amoeba, and for food vacuole it is present in protozoans, right? So if that is true in both the cases, for in protozoan also contractile vacuole is present, in amoeba also food vacuole is present, both are formed in both the cases. So but in NCERT particularly it is given, it is present in protozoans and it is present in amoeba, right? Next is functions of vacuole. Now what are the functions which are performed by vacuole? It is number 1, maintenance of osmotic balance, means osmoregulation. Osmoregulation means balance of water and solid particle, right? Next is function is storage of waste material, and it is also used for storage of some pigment, that is anthocyanin. Anthocyanin is a pigment which gives color to the petals, right? Next, it provides rigidity to the plant cell. So these are the basic functions of vacuole which is performed in plant cell, and type of vacuole is important. One is food vacuole, contractile vacuole, and sap vacuole. So these are all about endomembrane system in which four organelles are involved, that is endoplasmic reticulum, Golgi apparatus, vacuole, and lysosome. Next we will be discussing about mitochondria and plastids. So next organelle to be discussed is mitochondria. So in next lecture we'll discuss about mitochondria, plastid, and ribosomes.